There's a microbe, a single-celled organism. That's so powerful, so transformative, it rewrites what we thought we knew about health, immunity, and even mental function. It's not a virus that causes disease. It's not a bacterium we rush to kill. In fact, it's a microbe we're just beginning to truly understand, one that may hold the key to unlocking chronic disease prevention, metabolic health, immune regulation, and emotional well-being. And yet, this microbe doesn't live in your bloodstream, in your lungs, or on your skin. It lives in your gut. More precisely, it lives in your colon, part of the vast community we now call the human microbiome. What we once thought of as passive passengers in our digestive tract are now being recognized as vital regulators of health. Among these microbes, one in particular stands out, Akkermansia muciniphila. This single organism is proving to be one of the most important predictors of health in modern research. Let's start with the basics. Akkermansia muciniphila is a mucin-degrading bacterium. That means it thrives in the mucus layer that lines your intestines, not just living in it, but feeding off it. And counterintuitively, by feeding on this mucus, it stimulates your body to produce more of it, maintaining a healthy intestinal barrier. Why does that matter? Because your intestinal barrier is the first line of defense between your bloodstream and the contents of your gut. When that barrier breaks down, a condition often referred to as leaky gut, bacterial fragments, toxins, and undigested food particles can leak into the bloodstream. That sets off a cascade of immune responses, chronic inflammation, and contributes to diseases like obesity, type 2 diabetes, autoimmune conditions, and even neurodegeneration. Now, how do we know that Akkermansia muciniphila plays this protective role? We have both human and animal studies. In obese mice, increasing the population of Akkermansia led to improved insulin sensitivity, reduced fat mass, and a dramatic drop in inflammation. In human studies, people with higher levels of this microbe have healthier blood sugar regulation, lower cholesterol, better weight control, and less systemic inflammation. But it doesn't stop at metabolic health. Emerging research suggests this microbe is even involved in immune modulation, influencing how the body responds to cancer therapy and infection. In fact, patients undergoing immune checkpoint inhibitor therapy, drugs that help the immune system attack cancer, respond better when their gut contains abundant Akkermansia muciniphila. When it's absent, the treatment often fails. That tells us something profound. The effectiveness of our most advanced immunotherapies may not depend only on the drug, but on the microbes living in our colon. But the implications go even further. Consider the brain. We now understand that the gut and brain communicate constantly through what's known as the gut-brain axis. Short-chain fatty acids, chemicals produced when gut bacteria ferment dietary fiber, have direct anti-inflammatory effects on the brain and may reduce the risk of depression, anxiety, and even neurodegenerative diseases like Parkinson's and Alzheimer's. And Akkermansia muciniphila is one of the main producers of these short-chain fatty acids. That means your mood, your clarity of thought, your neurological health. They're all being quietly influenced by the balance of microbes in your gut, especially this one. Now, the question becomes, how do we cultivate Akkermansia muciniphila? The answer, thankfully, is rooted not in pharmaceuticals, but in lifestyle. Fasting is one powerful stimulus. Time-restricted eating or intermittent fasting has been shown to increase the relative abundance of Akkermansia. Polyphenol-rich foods like cranberries, pomegranates, green tea also help promote its growth. Prebiotic fibers such as inulin-resistant starch and mucilaginous foods like okra and flax seeds can nourish it indirectly. And there's even evidence that metformin, a common diabetes drug, may partially work by fostering this bacterium. But perhaps the most extraordinary part of this story is what it reveals about us, the human organism. We are not solitary beings. We are ecosystem superorganisms. For every human cell, there are at least 1 to 10 microbial cells. For every human gene, there are hundreds of microbial genes influencing our metabolism, our hormones, and even our thoughts. When we live in harmony with these microbes, when we feed them well and avoid what harms them, like ultra-processed foods, emulsifiers, artificial sweeteners, and unnecessary antibiotics, they reward us with resilience, vitality, and clarity. The most powerful medicine may not be in a capsule, but in a combination of food, time, and nature. A rhythm of life that respects not just the human body, but the microbial universe within it. The microbe that changes everything isn't just Akkermansia muciniphila. It's the understanding that our health is inseparably linked to the smallest organisms on Earth. And that through small, consistent changes in how we eat, move, and live, we can profoundly shift the trajectory of our health, not just for today, but for decades to come. Akkermansia muciniphila is a bacterium that resides within the mucus layer of the intestines, and its role in human health is far more profound than scientists previously imagined. Rather than being a passive resident, this microbe actively participates in maintaining the integrity of the intestinal barrier, which is a vital line of defense between the external environment within the gut and the sterile internal environment of the bloodstream. The structure of the intestinal barrier includes epithelial cells tightly linked together, covered by a layer of mucus secreted by goblet cells. This mucus acts as a physical and immunological shield, preventing harmful pathogens, bacterial components, and undigested food particles from entering systemic circulation. Akkermansia muciniphila thrives within this mucus layer, and while it may seem paradoxical, it feeds on the very mucus it inhabits. However, rather than depleting this protective layer, its activity stimulates the body to produce more mucus, thereby reinforcing the barrier rather than breaking it down. This dynamic relationship is one of balance and renewal. The breakdown of mucus by Akkermansia appears to signal epithelial cells to upregulate the production of mucins, the proteins that form the mucus layer. This feedback loop ensures the mucus remains thick, resilient, and constantly renewed. A healthy mucus layer limits the translocation of lipopolysaccharides, LPS, and other inflammatory bacterial components into the bloodstream. These molecules, when absorbed systemically, are known triggers of low-grade chronic inflammation, a condition closely tied to insulin resistance, obesity, cardiovascular disease, and neurodegenerative disorders. Therefore, the integrity of the gut barrier, bolstered by Akkermansia muciniphila, is a central pillar in preventing chronic metabolic and inflammatory conditions. Several studies have shown that individuals with higher levels of Akkermansia have significantly lower markers of systemic inflammation. For example, C-reactive protein CRP and interleukin-6L6F, both major indicators of inflammation, are found in lower concentrations in people with abundant Akkermansia. This is not merely a correlation. Mechanistic animal studies have demonstrated that introducing Akkermansia into the gut of obese or metabolically impaired mice reduces gut permeability, lowers inflammation, and improves overall metabolic profiles. These findings have been replicated across different experimental models, reinforcing the idea that this microbe plays a causal role in enhancing gut barrier function and modulating immune activity. One of the mechanisms by which Akkermansia achieves this is by increasing the expression of tight junction proteins. These proteins, such as occludin and claudins, seal the spaces between adjacent intestinal cells, making the epithelial barrier more robust and less susceptible to disruption. In conditions where these proteins are downregulated, such as in cases of dysbiosis, high-fat diets, or chronic stress, the gut barrier becomes compromised. This results in what is often referred to as leaky gut, a state where the intestinal lining allows microbial components and toxins to seep into circulation, triggering systemic immune responses. By supporting tight junction integrity, Akkermansia muciniphila serves as a protective agent against this breach. Furthermore, this microbe influences the production of anti-inflammatory compounds within the host. It has been shown to interact with Toll-like receptors on epithelial and immune cells, leading to a more balanced immune response that avoids overactivation. Unlike pathogenic bacteria that incite a strong inflammatory reaction, Akkermansia seems to train the immune system to remain alert yet not overreactive. This modulation helps to prevent inappropriate immune responses that can lead to autoimmune conditions or chronic inflammatory states. The balance it promotes between pro and anti-inflammatory signals is essential for immune homeostasis. Additionally, Akkermansia muciniphila contributes to the production of short-chain fatty acids, especially acetate and propionate, through its fermentation of mucins and other substrates. These fatty acids are known to nourish colonocytes, the cells lining the colon, thereby maintaining epithelial health. They also serve systemic roles influencing appetite regulation, insulin sensitivity, lipid metabolism, and inflammation. The presence of these compounds further strengthens the gut lining and creates an environment less conducive to the overgrowth of harmful bacteria. This microbial competitiveness is another layer of protection, as a diverse and balanced microbiota resists colonization by pathogens and opportunistic organisms. In light of these functions, Akkermansia is increasingly being explored as a probiotic candidate or as part of next-generation therapies for chronic disease. Pilot human trials have already begun, with pasteurized forms of Akkermansia showing promise in reducing insulin resistance and improving liver function in people with metabolic syndrome. What makes this microbe so unique is its dual ability to act both structurally, by reinforcing the gut barrier, and functionally, by regulating immune and metabolic processes. Its presence is not merely a biomarker of health, it is an active contributor to it. The very fact that it feeds on mucus, a substance once thought of simply as waste or protective goo, reveals the intricate and symbiotic relationships that exist between the human host and its microbial inhabitants. As our understanding deepens, it becomes clearer that restoring or preserving levels of Akkermansia muciniphila may become a frontline strategy for the prevention and management of a wide range of chronic diseases rooted in inflammation and gut dysfunction. Higher levels of Akkermansia muciniphila are strongly associated with improved insulin sensitivity and overall metabolic health, marking it as one of the most promising microbial indicators of metabolic function in humans. This bacterium resides predominantly in the mucus layer of the gut and has a unique ability to interact with the host's metabolic pathways in ways that go far beyond digestion. Its presence correlates with better control of blood glucose, healthier lipid profiles, reduced visceral fat, and improved regulation of body weight. These associations are not just observational, They have been confirmed in both human and animal studies, and increasingly, mechanistic pathways are being identified that show how Akkermansia communicates with host tissues to regulate energy balance and metabolic efficiency. In people with type 2 diabetes or insulin resistance, levels of Akkermansia muciniphila are often significantly reduced. This inverse relationship suggests a potential causative role of its depletion in the development of metabolic syndrome. Several controlled studies have administered either live or pasteurized Akkermansia to subjects with metabolic disorders and have observed improvements in insulin sensitivity, reductions in fasting glucose, and even decreases in markers of liver dysfunction such as ALT and AST. These benefits appear to occur independently of significant changes in body weight, indicating that Akkermansia exerts its effects through specific metabolic signaling pathways rather than simply via weight reduction. One key mechanism through which Akkermansia improves insulin sensitivity is by reducing systemic low-grade inflammation. Chronic inflammation, often driven by increased intestinal permeability and metabolic endotoxemia, impairs insulin signaling at the cellular level. By strengthening the gut barrier and preventing the translocation of inflammatory microbial components, such as lipopolysaccharides, into the bloodstream, Akkermansia helps maintain a metabolic environment that is conducive to proper insulin signaling. Inflammation in adipose tissue and the liver is particularly damaging, as it interferes with glucose uptake and insulin receptor function. The anti-inflammatory effects of Akkermansia extend to these organs, allowing them to respond more effectively to insulin and manage blood glucose more efficiently. Additionally, Akkermansia influences the secretion of gut-derived hormones that regulate satiety and glucose metabolism. One such hormone is glucagon, like peptide-1 or GLP-1. This hormone is released from intestinal ELP cells in response to food intake and enhances insulin secretion while suppressing glucagon release. It also slows gastric emptying, prolonging feelings of fullness and preventing postprandial glucose spikes. Studies have shown that the presence of Akkermansia muciniphila is associated with higher levels of GLP-1, suggesting that this bacterium plays a role in regulating the hormonal environment of the gut in favor of metabolic balance. Beyond hormone secretion, Akkermansia also contributes to the metabolism of dietary polyphenols and fibers, converting them into bioactive compounds that influence insulin sensitivity and lipid metabolism. When it ferments mucins and dietary substrates, it produces short-chain fatty acids such as acetate and propionate. These metabolites not only serve as energy sources for colonocytes, but also enter the circulation and interact with receptors in the liver, muscle, and adipose tissue. Propionate, for instance, has been shown to inhibit cholesterol synthesis and improve glucose tolerance. Acetate can modulate appetite, regulating centers in the brain, influencing energy intake and helping to reduce the risk of overeating and weight gain. These effects combined lead to a systemic improvement in the metabolic profile of the host. One of the more remarkable aspects of Akkermansia's role in metabolic health is its potential in reversing or mitigating the effects of high fat or high sugar diets. In experimental animal models fed with obesogenic diets, supplementation with Akkermansia has resulted in reduced fat accumulation, improved glucose homeostasis, and restoration of gut barrier function. Even in the context of continued dietary stress, this microbe exerts protective effects that buffer against the worst outcomes of poor dietary choices. It does not replace the need for dietary improvement, but it does indicate that microbial interventions can powerfully augment lifestyle measures and offer support where dietary compliance is inconsistent. Moreover, the metabolic benefits of Akkermansia are being explored in the context of liver diseases such as non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. These conditions are closely tied to insulin resistance and metabolic syndrome, and Akkermansia has shown promise in reducing hepatic lipid accumulation, inflammation, and fibrosis in both preclinical and early human studies. This suggests a broader role for the microbe beyond the intestine, influencing systemic metabolism through both direct microbial-host interactions and through the mediators it produces. Another layer of influence is the interaction between Akkermansia and other beneficial gut microbes. By contributing to a healthy gut environment through mucus renewal and short-chain fatty acid production, Akkermansia promotes the growth of other commensals that work synergistically to regulate metabolism. These include bifidobacteria and certain firmicutes that participate in fiber fermentation and help maintain a balanced microbial ecosystem. This microbial cooperation enhances metabolic outcomes and adds to the resilience of the host against metabolic disease. What emerges from the research is a picture of Akkermansia as not just a passive marker of health, but an active player in metabolic regulation. Its abundance reflects and influences a host of interconnected systems, from gut integrity and immune signaling to hormone release and nutrient metabolism. This makes it a compelling target for future metabolic therapies, especially in the context of rising global rates of obesity, diabetes, and related disorders. By restoring Akkermansia levels through diet, lifestyle, or microbial supplementation, individuals may be able to reestablish metabolic harmony and reduce their risk for a wide spectrum of chronic conditions rooted in insulin dysfunction and metabolic imbalance. The presence of Akkermansia muciniphila in the gut microbiome has been linked to enhanced immune responses and improved outcomes in cancer therapy. particularly with the use of immune checkpoint inhibitors. These treatments, such as anti-PD-1 and anti-CTLA-4 therapies, are designed to reactivate the immune system's ability to recognize and attack cancer cells. However, their efficacy varies widely among patients, and recent research has identified the composition of the gut microbiota as a significant factor influencing treatment success. Akkermansia muciniphila has emerged as one of the key microbial species associated with positive responses to these immunotherapies. Patients whose gut microbiomes are rich in Akkermansia are more likely to experience tumor shrinkage and longer survival times compared to those with depleted levels. Mechanistically, Akkermansia appears to prime the immune system in a way that enhances the activity of cytotoxic T cells, which are essential for attacking cancer cells. It does this by modulating the host's immune landscape, particularly within the tumor microenvironment. In studies involving germ-free or antibiotic-treated mice, the introduction of Akkermansia into the gut led to improved recruitment and activation of CD80+ T cells in tumor sites. These immune cells are central to the body's anti-cancer defense, and their presence in higher numbers often correlates with better treatment outcomes. The microbe also influences the balance of T helper cells and regulatory T cells, skewing the immune response toward a more pro-inflammatory, tumor-fighting state. A further evidence of Akkermansia's impact on cancer therapy comes from fecal microbiota transplantation (FMT) studies. When stool from cancer patients who responded well to immunotherapy was transplanted into germ-free mice, those mice also responded better to cancer treatment. Intriguingly, one of the consistent differences in the microbial composition versus non-responder patients was the abundance of Akkermansia muciniphila. Supplementation with this bacterium alone was enough to restore responsiveness to immune checkpoint therapy in animal models, even without the full microbial diversity of the original donor. This highlights the uniquely potent role of Akkermansia in modulating host immunity and sensitizing the body to advanced treatments. Akkermansia also interacts with antigen-presenting cells, particularly dendritic cells, enhancing their ability to process and present tumor antigens to T cells. This is a critical step in the immune activation cascade. Without proper antigen presentation, T cells remain ignorant of the tumor's presence. By boosting dendritic cell function, Akkermansia increases the likelihood that cancer cells will be recognized and targeted. In addition, it has been shown to increase the expression of co-stimulatory molecules and pro-inflammatory cytokines, such as IL-12 and IFN-gamma, which are key mediators of effective anti-tumor immunity beyond cancer therapy. Akkermansia's role in immune regulation extends into infection resistance and autoimmune disease prevention. Its presence correlates with a well-balanced immune system that is both responsive to threats and tolerant of non-harmful stimuli. This is particularly important in an era where immune dysregulation relies not only cancer progression but also chronic inflammatory and autoimmune diseases. Akkermansia appears to support mucosal immunity by maintaining the integrity of the intestinal lining and promoting the production of secretory IgA, an antibody that neutralizes pathogens at mucosal surfaces. This provides the first line of immune defense without inciting excessive inflammation. In conditions like multiple sclerosis, inflammatory bowel disease, and systemic lupus erythematosus, studies have shown reduced levels of Akkermansia in affected individuals. This depletion may contribute to the loss of immune tolerance and the breakdown of gut barrier function, both of which are central features of autoimmunity. Reintroducing or stimulating the growth of Akkermansia may help restore immune equilibrium by promoting regulatory T cell activity and preventing inappropriate immune response against self-tissues. The influence of Akkermansia on immunity is also mediated through its metabolic byproducts. The short-chain fatty acids it produces, especially acetate and propionate, have well-established roles in regulating immune cell differentiation and function. These compounds can enter systemic circulation and act on distant tissues, including bone marrow and lymphoid organs, to influence the development and behavior of immune cells. For instance, acetate has been shown to enhance the function of macrophages and neutrophils in clearing infections, while propionate can modulate between pro-inflammatory and regulatory immune cells in the context of viral infections. Akkermansia has also shown promise. Some early data from COVID-19 studies suggest that patients with higher levels of Akkermansia muciniphila had less severe disease and better immune responses. While this area requires more exploration, the preliminary findings align with broader evidence that this microbe supports immune readiness and resilience under stress. Its role in reducing inflammatory responses while preserving effective immune activation makes it a critical component of a well-functioning immune ecosystem. Another important layer is the effect of diet and lifestyle on Akkermansia's abundance, and consequently on immune function. Nutritional strategies that support Akkermansia growth, such as diets high in polyphenols, fiber, and fasting periods, may offer indirect ways to enhance immune surveillance and treatment efficacy. Conversely, factors that deplete Akkermansia, including high-fat Western diets, artificial sweeteners, and chronic antibiotic use, could undermine immune defenses and contribute to treatment failure in cancer or heighten susceptibility to infection. This connection between gut health, microbiome composition, and systemic immunity is reshaping the future of personalized medicine, particularly in oncology and immunotherapy. Akkermansia muciniphila plays a critical role in producing short-chain fatty acids as schizphus, particularly acetate and propionate, which directly support brain and neurological health through the gut-brain axis. This communication pathway between the gastrointestinal system and the central nervous system is highly complex and is mediated by neural, hormonal, and immunological signals. The gut microbiota, and specifically Akkermansia, influence these signals by generating bioactive metabolites that cross into systemic circulation and reach the brain, where they modulate inflammation, neurotransmitter activity, blood-brain barrier integrity, and even neurogenesis. This microbial neural interaction highlights a significant shift in our understanding of mental and neurological health, linking mood, cognition, and neurodegenerative disease risk to the presence or absence of specific microbes in the gut. Short-chain fatty acids, the primary metabolic products of Akkermansia, are produced through the fermentation of mucins and complex carbohydrates. These molecules are not only local energy sources for the cells lining the colon, but are also signaling molecules with systemic effects. Acetate and propionate can cross the blood-brain barrier, where they influence neuroinflammation and neurotransmission. Studies have demonstrated that these schifas increase the production of brain-derived neurotrophic factor, BDNF, a key molecule involved in learning memory and synaptic plasticity. Reduced levels of BDNF are associated with depression, anxiety, and neurodegenerative conditions such as Alzheimer's disease. By supporting BDNF expression, Akkermansia indirectly contributes to healthier brain function and resilience under psychological stress. Another way Akkermansia impacts neurological health is through its role in regulating inflammation. Neuroinflammation is now recognized as a central component of many psychiatric and neurodegenerative disorders. Microglia, the immune cells of the brain, respond to peripheral inflammatory signals, especially those originating from the gut. When the intestinal barrier is compromised and bacterial endotoxins such as lipopolysaccharides (LPS) leak into the bloodstream, systemic inflammation can trigger microglial activation and brain inflammation. Akkermansia helps prevent this cascade by maintaining gut barrier integrity, reducing LPS translocation, and limiting peripheral inflammatory responses that would otherwise reach and disrupt the brain's internal environment. Additionally, is produced by Akkermansia bind to specific protein-coupled receptors GPAR41 and GP43 located on immune and nervous system cells. This binding modulates the release of cytokines and neurotransmitters, resulting in reduced expression of pro-inflammatory markers and enhanced synthesis of serotonin and dopamine precursors. Since the majority of serotonin is produced in the gut, and its signaling is deeply intertwined with mood regulation, this microbial modulation has significant implications for mental health. In fact, altered gut microbiota compositions, particularly those low in Akkermansia, have been observed in individuals with depression, anxiety disorders, and autism spectrum conditions. In addition to protecting neurons from oxidative damage, selenium supports neurotransmitter balance, which is essential for mood regulation and emotional resilience. Dopamine, serotonin, and norepinephrine are all influenced by oxidative and inflammatory states in the brain. When oxidative stress becomes chronic, it disrupts the synthesis and receptor sensitivity of these neurotransmitters, leading to symptoms of anxiety, depression, and cognitive fog. Selenium's antioxidant properties help stabilize the environment in which these neurotransmitters operate, supporting healthy mood and mental clarity. Clinical studies have shown that individuals with low selenium levels are more likely to experience depressive symptoms, irritability, and fatigue. In one notable study, participants who consumed just one Brazil nut per day for 12 weeks experienced significant improvements in mood and reductions in anxiety. This change occurred without the use of pharmaceuticals and was attributed to the normalization of selenium levels and the associated increase in glutathione peroxidase activity. The participants did not change their diets or lifestyles otherwise, which points to the remarkable effect of correcting a single micronutrient deficiency. This study highlights the possibility that mental health symptoms, sometimes attributed to complex causes, may be influenced in part by subtle nutritional imbalances, particularly involving trace minerals like selenium. Selenium's neuroprotective functions are not limited to older adults. In developing children and young adults, adequate selenium is critical for proper brain development and neurological function. Animal studies have demonstrated that selenium deficiency during key developmental windows can result in lasting impairments in memory, learning, and motor coordination. This underscores the importance of ensuring sufficient selenium intake across the entire lifespan, not only to prevent decline but also to support optimal brain growth and function. Another emerging area of research involves selenium's interaction with the blood-brain barrier, a selectively permeable membrane that protects the brain from pathogens and toxins while allowing essential nutrients to pass through. Selenium appears to play a role in maintaining the integrity of this barrier by supporting the function of endothelial cells and reducing inflammation that might otherwise compromise its structure. A weakened blood-brain barrier is a feature of many neurological disorders, including multiple sclerosis and Alzheimer's, and selenium's ability to preserve its integrity may offer an additional layer of protection beyond individual brain cells and structures. Selenium influences the broader neural networks that govern behavior, memory, and cognition. Functional MRI studies have shown that oxidative stress can impair communication between brain regions, reducing the efficiency of neural processing and decision-making. By reducing this oxidative burden, selenium helps preserve network connectivity and signal transmission speed, both of which are crucial for maintaining high-level cognitive function. This is especially important for individuals under chronic stress, which increases oxidative stress and can impair brain function even in the absence of structural disease. Brazil nuts offer a natural, food-based solution to boost selenium levels without the risks associated with synthetic supplementation. Just one nut contains enough selenium to meet or exceed the daily requirement for most adults, providing a simple way to enhance neurological protection through diet. Because selenium in Brazil nuts is bound to amino acids, it is readily absorbed and utilized by the body, making it a reliable source for supporting brain health over time. This aligns with a growing body of evidence that nutrition plays a foundational role in neurological resilience, and that even small dietary changes can yield meaningful improvements in cognitive function and emotional well-being. Selenium plays a vital role in DNA protection and cellular repair system, making it a key player in cancer prevention and long-term cellular health. One of the primary ways selenium contributes to DNA integrity is through its support of antioxidant enzymes that reduce oxidative damage to genetic material. Every day, cells are exposed to oxidative stress from environmental toxins, inflammation, metabolic processes, and even normal cellular respiration. This oxidative stress leads to the formation of free radicals, which can damage DNA strands and increase the likelihood of mutations during cell division. Selenium, as an integral component of glutathione peroxidase and other selenoproteins, helps neutralize these free radicals and prevent them from causing harm to DNA. Maintaining DNA stability is critical because when mutations accumulate, they can disrupt cellular function and potentially initiate the process of tumor formation. Another important mechanism by which selenium supports cellular health is through its influence on apoptosis, or programmed cell death. Apoptosis is the body's natural way of removing damaged or potentially cancerous cells before they can proliferate uncontrollably. In healthy tissues, apoptosis acts as a protective filter, identifying cells with abnormal DNA or malfunctioning mitochondria and directing them towards safe elimination. Selenium enhances this process by modulating the expression of genes and proteins involved in apoptosis, including p53 and caspases. By supporting effective apoptosis, selenium helps the body eliminate cells that might otherwise contribute to cancer development. This is especially important in tissues with high turnover rates, such as the skin, gastrointestinal tract, and immune system, where constant cell division creates more opportunities for errors to occur. Several studies have linked low selenium levels with an increased risk of various cancers, including prostate, lung, colorectal, and liver cancer. One of the most prominent trials in this area, the Nutritional Prevention of Cancers (NPC) study, found that selenium supplementation significantly reduced the incidence of prostate, colon, and lung cancers in individuals with initially low selenium status. The underlying reason may be selenium's dual action of reducing DNA damage and supporting immune surveillance mechanisms that detect and remove early cancerous cells. Additionally, selenium influences angiogenesis, the formation of new blood vessels, which tumors rely on to grow and spread. By modulating angiogenic signaling pathways, selenium may restrict a tumor's ability to create its own blood supply, thereby limiting its growth potential. Selenium also supports enzymes that participate in DNA repair processes, such as those responsible for base excision repair and nucleotide excision repair. These systems are crucial for correcting single-strand breaks and removing damaged DNA bases that could otherwise result in mutations. Selenoproteins contribute to a redox environment that allows repair enzymes to function optimally, ensuring the fidelity of DNA replication and transcription. Without sufficient selenium, these repair mechanisms can be impaired, increasing genomic instability and raising the risk of carcinogenesis. In this way, selenium acts not just as a preventive agent, but as a continual safeguard that preserves genomic accuracy with each cycle of cell division. Furthermore, selenium plays a role in regulating cellular proliferation. It influences signaling pathways such as the mitogen-activated protein kinase (MAPK) pathway and the nuclear factor kappa B (FB) pathway, both of which are involved in cell growth, inflammation, and immune responses. Dysregulation of these pathways is a hallmark of cancer, as they promote unchecked cellular proliferation and resistance to apoptosis. By modulating these pathways, selenium helps maintain a balance between growth and inhibition, preventing the overactivation of processes that could lead to tumor formation. In addition to its cellular-level effects, selenium has a broader role in supporting immune surveillance, the body's ability to detect and eliminate abnormal or cancerous cells. Natural killer NK cells and cytotoxic T cells are critical components of this defense system, and their activity is closely tied to selenium status. Selenium enhances the ability of these immune cells to recognize and destroy cells exhibiting abnormal antigens or signs of mutation. This relationship between selenium and immune competency further reinforces its role in early cancer defense and tumor suppression. Brazil nuts, due to their exceptionally high selenium content, offer a natural way to support these protective mechanisms. By providing organic forms of selenium such as selenomethionine, Brazil nuts contribute to the incorporation of selenium into selenoproteins that perform these critical functions. Studies have shown that regular consumption of Brazil nuts can raise plasma selenium levels and enhance the activity of antioxidant enzymes within weeks. This translates into greater DNA protection, improved immune function, and more efficient removal of potentially malignant cells before they can form tumors. Selenium's role in hormone-sensitive cancers such as prostate and breast cancer has also drawn attention. Hormonal fluctuations can influence oxidative stress and cellular proliferation in hormone-sensitive tissues, creating an environment more prone to mutation and growth disturbances. Selenium helps to counteract this through its antioxidant effects and ability to regulate hormone metabolism, especially in conjunction with other nutrients such as vitamin E and zinc. These synergistic interactions highlight how selenium operates within a network of micronutrients that collectively support anti-cancer defenses and hormonal balance. In the context of chronic inflammation, which is a known risk factor for cancer development, selenium plays a key role in reducing inflammatory cytokines and blocking signaling pathways that promote cellular transformation. Chronic inflammation creates a microenvironment rich in growth factors, free radicals, and inflammatory mediators, all of which contribute to the development of cancer. By reducing inflammation at the cellular level, selenium disrupts this environment and makes it less conducive to malignant transformation. This multifaceted approach, reducing DNA damage, enhancing repair, promoting apoptosis, regulating immune responses, and decreasing inflammation, positions selenium as a crucial nutrient in the prevention and control of cancer-related processes throughout the body. Human health is deeply dependent on the microbiome, a vast and dynamic ecosystem of trillions of microorganisms that inhabit the body, particularly the gut. This collection of bacteria, viruses, fungi, and archaea performs functions that are so integral to our biology that is now considered an essential organ in its own right. Among these microbes, Akkermansia muciniphila has emerged as a keystone species, a microbe whose presence influences the structure and function of the entire microbial community, as well as the physiological processes of the host. The abundance or absence of Akkermansia reflects the broader health of the microbiome and offers insight into systemic processes that extend far beyond digestion. The microbiome as a whole contributes to metabolic regulation, immune function, neurochemical balance, inflammation control, and even the detoxification of harmful compounds. The presence of beneficial microbes like Akkermansia helps maintain this balance by competing with pathogenic bacteria, producing protective metabolites, and preserving integrity of the intestinal barrier. When this microbial balance is disrupted, a state referred to as dysbiosis, multiple systems within the body can become dysregulated. Conditions as diverse obesity, type 2 diabetes, cardiovascular disease, inflammatory bowel disease, autoimmune disorders, and neurodegenerative diseases have all been linked to disturbances in the microbiome. Akkermansia muciniphila specifically supports the structural foundation of the gut barrier by degrading mucins in a way that stimulates mucus renewal, thereby strengthening the protective layer that separates the intestinal lumen from systemic circulation. This process minimizes the leakage of microbial fragments like lipopolysaccharides into the bloodstream, which would otherwise trigger chronic low-grade inflammation. This inflammation is a known driver of insulin resistance, atherosclerosis, joint pain, and even cognitive decline. By reinforcing the gut lining and regulating immune signaling, Akkermansia indirectly shapes metabolic and neurological outcomes in ways that make its role disproportionately important compared to its population size. In addition to its structural contributions, Akkermansia plays a functional role in producing short-chain fatty acids such as acetate and propionate. These metabolites serve not only as energy sources for gut cells but also as signaling molecules that influence appetite, energy expenditure, fat storage, and insulin sensitivity. The regulation of these metabolic pathways is critical in a time when metabolic syndrome and its related conditions are affecting billions of people worldwide. The capacity of a single microbe to influence such broad human health speaks to the interconnectedness of microbial and human physiology. This interdependence becomes even more apparent in the context of immune regulation. Akkermansia's influence on immune balance helps prevent both underactive and overactive immune responses. It contributes to the maturation of immune cells, the regulation of pro- and anti-inflammatory cytokines, and the differentiation of regulatory T cells. A healthy level of Akkermansia in the gut has been associated with lower rates of allergic diseases, reduced severity of autoimmune flare-ups, and improved responses to vaccines and cancer immunotherapy. In each of these cases, the microbe acts not in isolation, but as part of a larger symphony of microbial interactions that collectively guide immune functions. That beyond immunity, the microbiome, with Akkermansia as a key player, exerts influence over the central nervous system through the gut-brain axis. Neurotransmitter production, inflammation, and neuroplasticity are all influenced by microbial metabolites and by the state of the gut barrier. A disrupted microbiome has been implicated in conditions such as depression, anxiety, autism, and Alzheimer's disease. Akkermansia, through its role in reducing systemic inflammation and producing brain-beneficial SEFs, contributes to the protection of cognitive function and mental health. These findings are reshaping our understanding of neurological disorders, suggesting that gut microbes are not merely bystanders but may be upstream modulators of disease progression or resilience. What distinguishes Akkermansia from many other microbes is its responsiveness to diet and lifestyle. Unlike pharmaceutical interventions, which often come with side effects or diminishing returns, microbial modulation through food, fasting, and physical activity provides a sustainable, low-risk strategy for improving health. Diets rich in prebiotic fibers and healthy fats have been shown to increase Akkermansia levels, while processed foods, artificial sweeteners, and chronic antibiotic use can deplete it. This makes it possible to directly influence one's microbiome, and by extension systemic health, through everyday choices. Interventions aimed at restoring Akkermansia levels may provide a powerful tool in personalized medicine prevention and long-term wellness. The implications of microbiome science extend into virtually every field of medicine. In oncology, immunology, endocrinology, gastroenterology, and psychiatry, the presence and activity of microbes like Akkermansia are becoming relevant biomarkers and therapeutic targets. Instead of viewing disease as an isolated dysfunction of a single organ or system, there is a growing appreciation for the microbiome's role in orchestrating health across multiple dimensions simultaneously. This systems-based understanding requires a shift in how health is defined and how interventions are designed. By recognizing the central role of the microbiome and the impact of keystone species like Akkermansia muciniphila, medicine can move toward approaches that are both preventive and deeply rooted in the body's natural biology from birth to old age. The microbiome evolves with us, influenced by delivery method, breastfeeding, environment, stress, diet, and medications. Akkermansia appears early in life and often declines with age, especially in those with chronic illness or frailty. Maintaining its presence may therefore be one of the keys to aging well, preserving metabolic flexibility, cognitive sharpness, and immune resilience. It is no longer sufficient to view health through the lens of human cells alone. Health is the product of a symbiotic relationship between host and microbiota, with Akkermansia as a central figure in this silent partnership that governs so much of human biology.